Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is how metabolomic profiling can unlock gene function and provide biological insight. Your presenter for today is Dr. Greg Michelotti. Dr. Michelotti is a biochemist with special expertise in the analysis of large metabolites and gene expression data sets. As principal investigator of a translational research laboratory at Duke University for 15 years, Dr. Michelotti successfully attracted both federal and pharmaceutical grants. His laboratory contributed to the identification of novel G-protein coupled receptor-mediated stress response cascades that contributed to deregulated epithelial growth responses. Collectively, this work resulted in the publication of over 70 peer-reviewed publications in high-impact journals, resulting in an H-index of 28. Dr. Michelotti joined Metabolon in 2015 to apply his expertise in signal transduction, cancer biology, and metabolism to assist investigators in the successful design and execution and analysis of large metabolomic data sets to provide biological insight into complex human pathologies. I'll be turning the presentation over to Dr. Michelotti now.
All right, thank you, Brandon. By way of overview, I'm going to introduce metabolomics, what our technology is, and how that's used for the precise quantification of biochemicals, and then how this information can be used to unlock existing omics data streams to help provide mechanistic insights into disease, as well as to understand the mechanistic basis and the basis of wellness and disease. By way of background of who we are, so Metabolon is a global leader in metabolomics, and our mission is to provide technology in advancing life sciences research and improving health. We were founded in 2000, over 18 years ago, and currently have over 200 employees worldwide with expertise across the spectrum of metabolomic profiling. This includes biochemistry, mass spectrometry, and software development. We operate a 54,000 square foot facility in the Research Triangle Park in North Carolina, where we're headquartered, and we run a CLIA and CAP certified and CAP accredited laboratory. So we're able to really run from discovery research to laboratory diagnostic tests. Collectively, we've completed over thousands of studies with clients across the research landscape, including academic, commercial, and government, that have collectively resulted in the publication of over 700 papers, 250 of which were last year alone. So what is metabolomics? For those of you, just to bring everyone up to speed, it's a technology for comprehensively and precisely measuring the perturbations in global metabolic profiles in biospecimens, and just as importantly, to interpret these findings to provide biological insights. I think we're all familiar on this slide with the central dogma of molecular biology, the flow of information from nucleic acids to proteins and ultimately to disease treatment or mechanism, what we call a phenotype. And nested in between these, juxtaposed to the phenotype or the disease treatment, are small molecules or metabolites. When I say small molecules, I mean typically biochemicals less than 1,000 Daltons. And they're uniquely positioned between the upstream information to integrate both genetic and non-genetic influences, including environment, lifestyle, and microbiome. So they really integrate upstream information to help provide insight into disease treatment and mechanism, providing actionable information into that. So while DNA and nucleic acids assess the risk of a disease and what genes might do for a disease outcome, metabolites really integrate this information and provide actionable information due to their proximity to the phenotype. Down below in the slide, you can see that there's a level of complexity with DNA, that there's 3 billion base pairs that have to be, we have to have information on. And while there are ongoing studies and we've generated large detailed data sets, and we're improving clinical significance, there's still a lot we don't know about how this information is used to result in treatment, and there's been a lot of information that is still to be gleaned. Conversely, metabolites, there's over 2,000 homeostatically maintained metabolites, the biochemistry of which is very well defined. We're resting on over 100 years of experimental knowledge to understand how metabolites are generated and how the pathways are connected. And as I said, they give a current health status due to their proximity to the phenotype and can provide an effective monitoring tool. So while we are resting on a lot of information of biochemistry, and it can integrate all the upstream genetic information and provide this actionable insight. So while we know a lot about these metabolites, there's still a lot in the genomic space that is still to be learned. There are vast swaths of unknown information, which is represented by this slide here. The genomic space represents 3.2 billion base pairs of information, and what we know is represented by the dots within that green space. So the coding space, or the coding proteins, are thought to represent a small fraction of that, about 64 megabytes, of which 25,000 known genes arise from there, and it's thought that about only 4,000 of those are associated with human disorders. Mutations of the human genome represent even less part of that space, representing about 4 million base pairs of information, about 130,000 kilobytes, or 130 kilobases are thought to be mutations with known human disorders, and 12 kilobytes of that information are thought to be mutations that actually result in a change in the protein sequence. So quite a bit of information still to be learned of genomics, despite the overwhelming amount of effort that's gone into genomic research. So clearly there's a lot that we need to know, and this has resulted in the identification of gene function for about 1,000 genes, for which we can identify the function of these genes. So clearly new and additional approaches are needed to help leverage this immense amount of genomic information to help identify what this information means and how it can impact human health. Sorry about that. And we believe metabolomics are one of the key approaches for putting all these pieces together and help leveraging the existing omics data streams to provide insight into human health. Shown here is how metabolomics can integrate the three factors I was discussing earlier: environmental influences, microbiota, as well as genetic influences. Because of their proximity to phenotypes, they integrate all this information and provide a real-time readout into human health status. Previous webinars have discussed microbiome research and the environment and how this has influenced metabolomics and metabolism. Today we'll be discussing DNA and genetic influences and how metabolomics can be used to leverage that information. But first, a brief overview of the platform of how we generate this information. Metabolon, as I said, invested a great deal of time and energy in the development of the precise measurement of metabolomics, and we do this from a standardized extraction from a biospecimen that is then run across a UHPLC mass spec, tandem mass spec platform in 4 methods, in both negative and positive electrospray ionization. And we run 4 methods to capture as many of the classes of biochemicals as we see to get the broadest coverage possible. And anyone who knows anything about mass spec knows that it can be a noisy platform, which can give rise to hundreds of thousands of ion features in each of the arms of the platform. Metabolon has invested significant intellectual and capital investments into deconvoluting these ions and to develop a chemocentric approach to eliminate noise, reduce data, and run these ion features against a validated chemical reference standard library of thousands of metabolites, thousands of standards, that results in the identification, the unequivocal identification of metabolites in a biospecimen. And this is predicated based on 3 key criteria: the assessment of retention time, accurate mass down to 1/10 of a thousandth of a Dalton, and then the mass spec fragmentation profile. And those 3 key criteria together collectively result in the accurate identification of metabolites that are present in the biospecimen. This then goes through exhaustive QA and QC so that the quality of the data is the highest possible. And that's on the technical side. On the biostatistics side and interpretation side, we have a team of biostatisticians here at Metabolon that are involved in the statistical analysis of data, typically a standard suite of tools, including high-level views of data analysis like hierarchical clustering, PCA analysis, random forest analysis, and bread-and-butter approaches for the analysis of the data to provide significant changes between groups in a given study. These data are then exported into Attractive formats, including heat maps, where we provide the information of all the metabolites detected within a study that are color-coded for significant changes up or down in comparison groups. And these metabolites that are detected are fully annotated in terms of the pathways that they belong to. This information can be graphically rendered or projected into pathway maps using our Metabolomic portal tool that investigators can have access to that provides a pathway projection of all the information and statistical analysis within the study. We also have a team of PhD level biochemists that provide interpretation for this information to provide biological relevance for what these changes mean in the context of the study. And this may be for biomarker discovery, mechanistic understanding, drug mode of action, for example, or changes in cellular characteristics. And as I said, this platform has been designed to be unparalleled to any metabolomics service providers in the field. And for this, we provide what's called a Tier 1 identification that is based on a chemical reference standard based on the 3 criteria I talked about earlier: retention time, accurate mass, and mass spec fragmentation profile that provides the most accurate identification of biochemicals that are present in a biospecimen. And this is relative to other metabolomics profile providers that typically provide less stringent level of certainty for their identifications that are Tier 3, Tier 4, or Tier 5 below this that are based on ion standards, so that every metabolite that is returned to investigators is returned with the highest degree of confidence and unequivocal identification of what these metabolites are with a precise level of quantification. And all this is to provide as comprehensive a survey of the metabolome as possible. This blue dots map represents all the metabolites that we can detect on our platform, and when analyzed over the course of a study to look at changes in biospecimens in study groups, for example, we're able to survey the entire metabolome and provide biological insight into perturbations in given pathways. And I think you can appreciate how all these pathways integrate and are connected. So again, we're resting on over 100 years of information of knowing how these metabolites interact and how they are correlating with each other. So how can this information be used to leverage existing omics data streams? And here I'll focus on DNA, the genetic component. And we've done some seminal work in this area where we asked the simple question: can metabolomics bolster findings in genomic studies? And here we worked with Carson Surrey. We collaborated with him on a study, as well as with Tim Spector, to see if we could functionally map variants in the genome of individuals to identify these changes or these variants with a biochemical step. That is, can we associate a change in a SNP to a change in a biochemical step? So plasma was taken from 2 distinct cohorts, over 8,000 subjects in total, the core cohort of German descent and the twins UK cohort of British descent. So 2 disparate populations that were collectively 8,000 subjects each, for which samples were taken and saturation SNP analysis was done. These same samples were then subjected to metabolome profiling here at Metabolon to determine if we could functionally map rare variants or provide information for what these rare variants may mean in these individuals. So what we're asking is that, can you find an allele with a frequency high enough in these 2 disparate populations in this sample size that has an effect high enough to result in a change in a biochemical step that we can detect? So with that tall order, the answer is absolutely yes. We were able to find these associations, that change in gene locus or genetic variant was able to be associated with a metabolic trait at a high enough significance that it survived a Bonferroni correction. So we're essentially able to functionally map a variant of unknown significance and show that it results in a change in a biochemical step. And these changes, or these SNPs that correlate with these metabolic steps, covered a very wide swath of metabolic pathways and functions. And these include 26 SNPs or gene variants that are associated with inborn errors in metabolism, 10 genes that are targets of drug targets and drugs in development, so providing insight into commercial development of how pharmacogenomics can work and how it can change biochemical steps and metabolism, 36 genes linked to complex diseases or drug responses, and then other aspects of genes that were involved naturally in enzymatic pathways or enzymes that change an enzymatic step that would naturally be associated with a metabolite, transporters that are involved in the transport of metabolites in and out of cells, as well as growth factors, ion channels, transcriptional regulators, as well as genes of unknown function that we're able to provide insight into what the function of these genes may be. So what that told us is that ultimately we're able to identify, let me go back here, there were over 80 SNPs that were found to be novel in this association, in this study. And when you think about 84 novel genes out of 104 metabolic loci associations, when we think about what earlier was discussed, that we roughly know the functions of about 1,000 genes. In one study, we're able to identify or functionally map the functions of 84 genes using this approach, adding approximately 10% to the previous genomic effort. It tells you that this is quite a powerful approach to help functionally map rare variants and leverage genomic information. And what that also tells you, given the relative size of the population and the number of metabolites we're able to detect, that we need a higher level of resolution to utilize the power approach, or better to leverage the power of this approach to identify more gene associations and functionally map more gene functions. And for that, we used the latest and greatest technologies, that is whole genome sequence that is linked to the latest iteration of our powerful Metabolon platform, which is now able to detect and quantify about 1,000 metabolites present in human blood. And for this, we used the Twins UK cohort. So 1,960 twins. Samples were collected at 3 separate time points, encompassing in total up to 2 decades. So 3 separate time points from the twin study were subjected to both whole genome sequencing and metabolomic profiling using our latest iteration of our precision metabolomics profile to map gene variants to metabolic levels. And this study was published just last year. One of the striking things that emerged from this is the remarkable heritability of the blood metabolome. So of 901 metabolites that were detected, 644 metabolites have consistent heritability. That is, that their levels did not change less than 2-fold or 50% over the 3 visits. And remember, these 3 visits encompass up to almost 2 decades, 18 years. So over this period of time, metabolite levels for 644 of the 901 metabolites were changing less than 2-fold. So there's a remarkable heritability to the genome. And then using the same association approaches, 246 metabolite associations were mapped to common and low frequency variants, common being a monoallele frequency of 5% and a rare frequency being less than 0.5%. And of this approach, 90 novel findings were identified. Again, significantly adding to the information of what are the function of some of these variants of unknown significance to functionally map these using metabolomic profiling, providing new insights and biomarkers to pathways and various diseases. And I know it's kind of confusing, but that polar bar plot shows the heritability of all of the pathways, super pathways of metabolomics. And the longer the bars, the more heritable the genome, or the metabolome. But ultimately, it was over 2/3 of the metabolites turned out to be stable in these populations in over 2 decades. I know this is kind of a busy slide, but this shows the Manhattan plot of the information and how the rare variants and the significant associated changes in metabolites with a biochemical a variant or mapped. And so while they're able to, this approach is obviously useful for the mapping of common variants of about 5% in the population. One of the approaches using this level of resolution with whole genome sequencing and our precision metabolomics profiling can also be used to look at rare variants, to functionally map these rare variants in the population with a minor allele frequency of less than 0.5%. And for this, to identify these, the authors took an approach to identify the metabolites or map the metabolites that are associated with these rare variants by looking at those that change from the population mean by over 4 standard deviations. So these are metabolites that are very largely changed relative to the population mean. And then to the detected metabolic abnormalities identified were the results of heterozygous rare variants that are commonly accepted as autosomal recessive diseases that are typically associated with inborn errors in metabolism. And to help assess the causality of this, they used 4 different criteria. One was the existence of unrelated individual outliers carrying the same variant. That is, does this same variant occur in other unrelated twins? The prediction of a functional role using protein structure analysis if the SNP or the genetic variant occurred in a protein coding sequence. Identification of additional independent rare variants in the same gene or metabolite association. And is there a gene harboring the variant that has been reported already with an associated inborn error metabolism? So these 4 criteria were used to help them establish causality of the metabolic gene variant association. And using this criteria, one of the top hits that came out was with dimethylglycine dehydrogenase, or DMGDH. This is a mammalian mitochondrial enzyme which plays an important role in the utilization of methyl groups from choline. So it's a flavin cofactor enzyme that essentially catalyzes the oxidative demethylation of dimethylglycine to form sarcosine. And 3 heterozygous variants were identified in 3 twin groups that are shown here in the middle part of the slide. And you can see from the genotype that these are heterozygous rare variants. And down below in the box plots and whisker plots, you see that the red dots on the left side show that these rare variants or these twins have dimethylglycine levels at much higher levels relative to the population mean. And in the middle and the right side of the panels, you can see how these levels are changed over the 3 visits, encompassing up to 2 decades in time, that they start elevated at the first time point and remain elevated over all 3 visits relative to the population mean. So using this approach, they're able to identify dimethylglycine levels that are associated with rare variants in dimethylglycine dehydrogenase gene. A second example of this is with the acyl-CoA dehydrogenase gene, or ACADs for short. So ACADs, point mutations, or SNPs, were associated with elevated levels of ethylmalonate, which is shown on the bottom left box plot. And you can see relative to the population mean that 2 variants resulted in exceedingly high levels of ethylmalonate resulting from this point mutation in the acyl-CoA dehydrogenase gene. And then further analysis of protein structural analysis and modeling showed that these point mutations were in the same regions on the middle left part of that for the point mutations in the study relative to established mutations that were reported in OMIM, that these were in the same neighborhood of these mutations and likely resulting in the causal increase in ethylmalonate levels as a result of perturbation of the genetic structure and then the protein structure. So they were able to map 2 functional rare variants using a metabolomic profile to show that these rare variants are associating significantly with a change in biochemical step to functionally map rare variants and provide genetic insight that was otherwise not able to be provided. So that provides an example of how to functionally map genetic variants. More broadly, new studies have shown that we can extend metabolomic profiling to show that it can correlate with other information as well. So not only from genetic scores, but also from polygenic scores that were derived from GWAS studies, as well as other omic streams like the microbiome, the proteome, as well as clinical laboratory measurements. And in a seminal paper that was published at the end of last year by Leroy Hood's group, this shows the clinical relevance of metabolomic profiling using the Pioneer 100 study. And here, 108 individuals were recruited across a wide range of ages, and samples were drawn, blood samples were drawn from individuals at 3 time points over 9 months. And high resolution density data were taken, personal data were taken from these individuals over these 9 months or at these 3 time points. Shown on the bottom part of the slide, over 218 clinical labs were taken. 643 metabolites were detected using our platform. Plasma proteome was detected using Olink technology, 262. Polygenic traits, so genetic traits that were associated with GWAS scores were compiled. 130 of those were tabulated for this study in these individuals, as well as the microbiome was assessed using 16S sequencing to assess operational taxonomic units, which essentially are proxies for microbial strains. And 4,616 of those were assessed. And the goal of the study was to identify how these omic streams correlated with each other. And this pretty figure shows a summary of this inter-omic correlation network, showing each of the nodes that I talked about on the previous page, clinical labs, genetic traits, proteins, microbiome, and a metabolome are color-coded. Each metabolite or analyte is representing a vertex or a point on that circle, and each line represents a significant pairwise correlation using adjusted p-value to adjust for multiple comparisons. So each of those lines represents a significant Spearman's correlation that was identified for each of the vertices on that circle. So that can be a pretty busy slide to look at. To help simplify or interpret these results, clusters or related measurements, or communities as they're called, were identified by pruning the network to remove edges with the highest betweenness. So this is using betweenness centrality approaches to help prune the network and clean the network to identify only the clusters of related measurements. And then this can be organized by physiologic analytes and their associated factors. So in this case, looking at polygenic scores correlate with blood analytes. So for example, the genetic risk of inflammatory bowel disease, or IBD, in Europeans and the European descent has been associated with about 110 SNPs. And this is these are published studies collectively. In this cohort, the polygenic score for irritable bowel disease from these 110 SNPs was significantly and negatively correlated with plasma cystine, which is the disulfide form of cysteine, which is, so that the correlation line plot is shown on the right side of the panel. And these values have been adjusted for age, sex, et cetera. And the shaded regions on there show the 95% confidence intervals for the slope of the line. So there's a negative correlation with cystine levels and the polygenic score established for inflammatory bowel disease. And in fact, previously published evidence provides evidence that plasma cystine and cysteine levels are abnormally low in patients with irritable bowel. And so this provides insight, if you know the cystine pathway, of how this, which is projected there on the left side of the panel, of what pathway it belongs to and its role in metabolic pathways. Excuse me. So this suggests the role for cystine and indeed oxidative stress in irritable bowel disease. So this helps uncover information in a previously thought to be healthy population of people that could potentially be at risk for developing diseases and early onset diseases using this approach. Now, the polygenic scores, of which there are 130 in this study, did not represent the vast majority of the analytes and the associations or correlations in the study. In fact, what you see here in panel A here of this slide shows that the metabolite and clinical lab correlations represented 97% of the correlations identified in the study, as might be expected, that metabolites in clinical labs would correlate highly. And as the genetic information is more vetted and the omics streams are more highly developed and added to this kind of approach, you can imagine that expands for the other omics streams, and genetic information would be a larger piece of that pie. But to provide some insight into other omics streams of how metabolomics can be used to leverage that information as well, using the same pruning approach to look at the cardiometabolic community in this case, a novel finding was shown that a gamma-glutamyl amino acid, in this case gamma-glutamyl tyrosine, was significantly associated or correlated with cardiometabolic clinical laboratory measures. And that is indicated by the red arrow showing where gamma-glutamyl tyrosine is, and then the purple lines emanating from there showing the significant correlations with a host of clinical chemistries. And that is encapsulated or summarized on panel C of that slide, showing that this associates with levels of P-adjusted values of glucose, insulin, LDL size, HDL cholesterol, triglycerides, as well as insulin resistance. And gamma-glutamyl tyrosine is produced by the enzyme gamma-glutamyl transferase, which is responsible for a large number of gamma-glutamyl derivative amino acids. But in this case, it's only the tyrosine derivative that came up as significant in this correlation. So while the enzyme gamma-glutamyl transferase is known as a biomarker of diabetes risk and liver dysfunction independent of BMI, this really hones down on a specific molecule, not just a class of molecules, a specific molecule that can be associated with these measures. And collectively, these results indicate that gamma-glutamyl tyrosine could be a candidate marker for diabetes risk independent of BMI. So this really helps expand our utility or information of assessing clinical measurements in the lab, how to approach individuals that may present with different variations of BMI or different variations of disease, and how they can be clinically managed in the best way, and how metabolome profiling can be a key step in that process. So in summary, I hope I've convinced you that metabolomics can be used to functionally map both common and rare genetic variants that provide novel insight into gene function, as well as potential role into health and disease. The metabolome provides a rich data set that reports on genetic traits and risk, the microbiome, and clinical laboratory measurements, and helps leverage that information to provide real-time insight into and mechanistic insight into human health and disease. The numerous correlations of the latter study of metabolites with clinical labs suggest that metabolomic information can complement or enhance clinical practice and or clinical research and advance the understanding of individual health and disease. And collectively, metabolomics illuminates large-scale genomic information through novel associations and the assignment of gene function. So we believe that metabolome profiling can be the key tool to help unlock existing omics data streams to really provide mechanistic insight into biological questions. Thank you, and I'm happy to answer any questions at this point.
We'll be taking a couple questions now, and in the event when we run out of time, we'll answer your question offline as soon as possible.
All right, so we have our first questions. How many samples are needed for adequate study power? That is a common question and a great question that we get. And I assume in this case that we're talking about human studies because this talk was based on population health and focused on human studies. But we do a full range of biospecimens across every imaginable source of biospecimens you could imagine, from blood to tissue to plants. So you can imagine there's quite a wide range of tissues that we analyze. In terms of study power for human studies, given inter-individual human variability, we recommend minimum sample sizes or group sizes of 25 in comparison groups. And this is based on the many years of expertise that we've developed in this space, as well as power analyses that we've done in these areas looking for specific effect sizes. So 25 is a minimum sample we recommend for human populations. With preclinical models, you can go down, especially with, if we're considering like black 6 mice, that we would recommend group sizes in the 6 to 10 range. And then with isogenic, like cell culture models, you can go down from there. So 5 samples per group, for example. So it gives you an idea of sample group numbers to appropriately and adequately power your study. One question I have is, can lipids be adequately analyzed? Yeah, so what I talked about here in this talk were global metabolomics, or the metabolites that we typically see or think of on a wall chart that are less than 1,000 metabolites. Of those, a large proportion of those are lipid classes, so polyunsaturated fatty acids, free fatty acids, eicosanoids and inflammatory lipids as well are all detected on this platform. We also have an ability that I didn't go into on our platform talk to analyze complex lipids as well, and that represents a separate workflow and a separate part of the platform, but we have the ability to provide absolute quantification for complex lipids as well, which will represent about another 1,000 metabolites in human blood that collectively between the global platform and the lipids platform for complex lipids, we can return or identify and quantify about 2,000 metabolites in circulation in humans. One final question that I think we have time for. Metabolomics and age-related macular degeneration. Well, it's not really a question. I assume you're asking if we can look at that. So we have looked at, you can imagine a vast array of diseases with the thousands of studies we've completed that we have experience in looking at a number of different human disease conditions. And in fact, we have looked at age-related macular degeneration, and in fact, there's been a paper on that using our technology that was published recently by Diba Hussain. I believe she's at Harvard, and she actually worked with a collaborator of ours, Jessica Lasky-Sue, in this project as well, where we're able to, in a relatively small sample population of about 25 individuals, I believe, we're able to identify changes in macular degeneration in a disease group versus a healthy cohort. In fact, they found that they're particularly perturbed in the people with the affliction of macular degeneration were deficient in glycerophospholipid metabolism. So we're able to provide significant mechanistic insight into potential issues relating to this from circulating metabolites. So similar approaches have proved equally fruitful in a wide variety of diseases, including diabetes, liver disorders, cardiometabolic disorders, microbiome-related disorders. So yes, we've been able to have significant success in these kinds of approaches.
Thank you for attending our webinar.